All right, we're up and running and recording. All right. Was everyone able to access the first exam on Monday? Any questions about the exam? Just to be clear on it, each one of the questions, I'm kind of looking for a high level conversational knowledge. Okay. You don't need to get into the weeds on things. So don't be anxious over it, you know. It's all everything that's on there is what we've covered in the past lectures over nervous systems and signaling systems or endocrine systems. If you need additional time, just let me know. We can ask Bean if that's okay to give you extra time. Pay attention. Anyway, so yeah, so take some extra time if you need it. Otherwise, they're going to be due on Monday. Just don't get behind with it because, as Val said, it all comes collected at the end, right? So, all right, so we are a little bit behind on the syllabus, but we'll catch up. I've This syllabus is designed, it has buffers in it so that we can kind of adjust accordingly for the semester. So we're going to jump into integumentary systems and developmental systems. So we'll talk about how occurs in insects today, and then on Monday as well. We'll continue that in chitin synthesis. And we'll talk quite a bit about different gene expression patterns and morphogens, as they're called, and how these different messenger RNA titers basically signal the anterior, posterior portions of a developing embryo, the dorsal ventral portions, and then how everything starts to kind of fine tune into like the individual segments or features of insects. So, like the mouth parts, the antennae, the wings, the legs, the abdominal segments, and so on. And how all that's regulated. What does an insect egg look like? Huh? It varies, doesn't it? It's not a simple structure, is it? It's quite complicated. Yeah. It's one of my favorite questions to ask to PhD students when they're taking their comprehensive exams. Go ahead. Right? Yeah. So go draw an egg on the board. And they always draw an egg, just an oval. But there's so many layers, right? There's so many, and they're all different types of characteristics, different features, different styles. I mean, eggs are just like they're amazing. Insect eggs are just amazing visually and structurally and how they appear and how they work. But that's what we're going to talk about today a little bit. And as I mentioned before, these morphogens or these concentration gradients of messenger RNAs and proteins and how they drive the different structural characteristics of that developing embryo to through its larval and its adult stages, and then those developmental patterns, of course, along the way. Hey, Ozzy, how are you? See if I can get rid of this thing here. I don't know if it's going to go away or not. All right. So as I mentioned before, insect eggs, they're a variety of different structural features, different shapes and sizes, different colors. But they have it's a critical structure, and that it creates this two-way barrier. So a variety of different things here that need to happen to be able to preserve or protect the integrity of that developing embryo. So, the insect egg, it prevents the loss of the egg contents to the environment. It reduces any type of disturbance of those egg contents to environmental hazards. It has to be flexible enough to pass through the ovipositor. It has to permit sperm flow to enter for fertilization purposes. It exchanges oxygen and carbon dioxide. And it also has to be built in a way that it facilitates easy escape of the larvae after embryogenesis. So there are multiple layers that make up insect egg membranes. Okay, so they do provide protective strategies, as I mentioned in the previous slide. They do form a barrier to laboratory manipulations for studying embryogenesis. it's quite complicated to be able to stick a needle or inject an insect egg. Okay. A lot of them are highly pressurized, so they'll explode. But to be able to surgically inject a needle to be able to alter different gene expression events or different embryogenesis or developmental activities is quite complicated. Mostly because there's also this self-repair. It does provide some ease for experimental injections, so you're not killing the embryo. It's somewhat permeable to different chemistries. You often have to dissolve the different layers that are shown over there in the right-hand side with different solvents to be able to allow certain chemistries to cross that barrier. Um, they have heat shock proteins that protect against denaturation, other environmental stressors. Eli, are you working with Leslie on the heat, on the temperature gradient project? Yeah, so she's interested in these heat shock proteins, right? That protect developing not only just the egg, but also the larvae and eventually the adults. And then, as I mentioned before, there are these stable gradients or these morphogens of messenger RNA titers, protein concentrations that guide the development of the embryo along the way. So you'll see these cross sections of the chorion or the eggshell of a Drosophila egg, and so you'll tend to have these ridges, different structures on the outside or the external part of that egg. You have the exochorion or the outer layer, most layer. You have a roof network. You have the outer endochorion, and they have these different pillars here that separate the different sections. You'll have this egg membrane. There's the wax layer that protects it from different chemicals and also desiccation. And then you have inside, you'll have the velatin envelope here, basically the egg proteins or for nourishment, and then the developing oocyte on the inner layer. Regardless of the egg or the insect, I should say, they all have these different structures. If you look at the cross section of these eggs, so these protective structures, but just know like the external part of it has a variety of different ridges and crevices and whatnot. But then you always have the exochorion and then the outer endochorion and then those pillars that help with that different network to protect the developing oocyte. So the eggshell, it's synthesized with yolk proteins after vitellogenesis or the production of these egg proteins. So you have follicle cells that surround the oocyte. We'll talk a little bit more about these here in a moment, but they surround the oocyte and they direct their products inward to secrete these different layers of the chorion or the eggshell. And then these subsequent layers are formed by apopposition. So the movement to different pole regions, whether it's the anterior, posterior, dorsal, ventral portions of the developing oocyte. And then there are these intercalation events in where the follicle cells overlap each other to form different layers. So during this choriogenesis event, again, it's a period of protein synthesis. And these proteins, again, act as morphogens or gradients to drive the head, the tail, the top, the bottom of that developing embryo. So there's a sequential activation of these genes and the movement of the proteins. Okay, so they're being activated at specific times, but then there's also that temporal activity to move them to different parts of the developing insect so that you can, or so that you're differentiating again the anterior, posterior, dorsal, ventral portions. And then there's a lot of these sulfur bonds or disulfide bonds that cross-link to harden that eggshell, again protecting the developing embryo. So as I mentioned previously, these follicle cells here, again, you'll have these different follicle cells here. They differentiate into border cells. You have stretched follicle cells. You have centripetal follicle cells, the main body cells, and then the posterior follicle cells. Basically, what's happening, as I mentioned in the previous slide, they're starting to differentiate the different portions of that developing embryo, okay, from or that oocyte, and basically protecting it, producing the posterior or the anterior portion, and then later on they'll start signaling for anterior ventral portions of that embryo. Yeah, Ozzy.
That broad thinking of the disulfide bond, does it function similarly to collagen? Because I know that has the crisscross.
Yeah, so a chitin would be another one too, and then they just kind of build on top of each other with those different bonding structures, right? And they're quite stable and hard to break, of course. So yeah, it's a good question. So if you look at the cross section of an insect egg prior to fertilization, you have this micropyle opening here, and this is synthesized from those border cells that I showed in the previous illustration. So it's nothing more than just a canal that leading to the oocyte, and it allows entry of sperm to that developing oocyte. You also have an operculum or a cap that surrounds the hatching regions. It's not really shown on here, but if you look at an egg, you'll get these kind of breaks in the chorion where it allows the larvae to escape during development. So it's just easy cracking points for escaping or exiting of the larva. But inside this whole process or this egg here, again, you're going to have localized maternal messenger RNA. We'll talk a little bit more about this in a moment here. You have the cytoplasm of the yolk, contains a lot of the organelles for the developing larva, the nucleus, of course, and then the chorion or the eggshell itself. So as I mentioned before, there are spatial and temporal gradients of gene expression that are occurring during this whole development event. And so you have vitelline genes that are expressed at stages 8 through 10. You have chorion genes that are expressed at stages 10 through 14, and then a variety of different proteins that undergo different post-translational modifications or relocations during development. So again, going back a couple of slides where those follicle cells start to distribute themselves in the anterior posterior portions. These vitelline genes here, again, these are earlier stage expressed genes that are producing vitelline for the embryo. And then later on, you get these chorion genes that are basically protecting the side of the leg or outside of the egg and giving it its structure. So again, really the takeaway here is that there are a cascade of different genes that are expressed both in time and space. So 3 broad questions here to take away from this section really at the end of the day. There are genes that are involved in spatial patterning of developing insects. There are polar gradients and pattern formation activities that establish the position of the developing insect. And then there are these homeotic genes that give the structures their key characteristics. Okay, the reason why head structures are different, wing structures are different, antennae, leg structures, abdominal segments. All of these different these genes play a role in those key characteristics for insect development. So in the first question here, the main genes that that are playing a role in the positioning or the spatial patterning, just the broad spatial patterning of that developing oocyte are bicoid and oscin. And these basically signal the posterior and anterior portions or orientation of that oocyte. But it starts with 2 other genes up here. You have a gherkin signal that's localized in the nucleus, and it's received by those posterior follicle cells that I showed you in a few slides previously. So this gherkin signal, or this protein, it binds to a torpedo receptor, and then it specifies the fate of these follicle cells as the posterior region the oocyte. These posterior follicle cells then send a signal back to the oocyte. It repolarizes the whole cytoskeletal framework of the oocyte. And then it starts localizing these oscar and bicoid messenger RNAs at either end of the oocyte. So what's happening here again, it's creating the broad separation or orientation of the anterior posterior portions of that oocyte. So once the cytoskeleton framework is created, this will cause the oocyte nucleus to move to the dorsal anterior part of the cytoplasm. And then again, you get a Gherkin signal that causes the nucleus or the follicle cells to express a KECON gene. And then this starts to form or establish the dorsal identity. Once the ventral cells, and then after the dorsal identity is formed, then the ventral cells here, they're not receiving that Gherkin signal. So then what they do is they express what is known as a pipe gene, and that's established in their ventral identity. Okay, so all insects express each one of these genes to be able to establish, again, the anterior, posterior, dorsal, ventral portions of the oocytes. So what about these polar gradients? So it's not just these 4 different genes. You have another 4 genes that are messenger RNAs that are working here to establish that position. So here in this graph, it shows an anterior oocyte and then the posterior oocyte. And then on the y-axis is the concentration of messenger RNAs. So the 2 messenger RNAs that establish the anterior orientation are hunchback and bicoid. So they're highly expressed in the anterior portion, and their concentrations decrease as we move to the posterior portion. And then caudal and nanos are highly expressed in the posterior portion, and then again, they're decreased in expression in the anterior portion. So these are the 4 primary messenger RNAs that encode those proteins for anterior posterior portioning or positioning of the oocyte. And then you have a series of these homeotic genes, and we'll break these down here in a moment. But basically, the homeotic genes differentiate the individual parts of the insect. Okay, so keep in mind what's happening with all these gene expression events are going from a broad kind of landscape design of that oocyte, and then they get fine-tuned into the specific features of the insect. Okay. However, homeotic genes are highly conserved across animals. Okay. And if they're highly conserved across insects, why do insects appear so different? Perfect. Yeah, it's the spatial and temporal expression of these genes that give different insect families, orders, their specific characters, right? Different species, genus, and whatnot, right? And so it's different expression patterns, but also different mutations will cause those. So there's a lot of work that's been done in this area in evolution developmental biology, okay, and embryogenesis is to go in and to manipulate these homeotic genes, and that you can find when they are being expressed and the concentration that they're being expressed and how they operate to produce a leg or an abdominal segment or a wing. Okay, so a lot of early site-directed mutagenesis of these homeotic genes will create flies that have legs coming out where their antennae are, wings coming out where their legs are. And so they move these around, these little Frankenstein bugs, to be able to identify how these homeotic genes operate and then what are going to be the structural features of the genes as they're expressed.
Can they do that by injecting mRNA into just different parts of the embryo, or is it?
You can do that. You can. The early work with site-directed mutagenesis is being able to clone out the specific homeotic gene and then manipulate that structure. right? So, or even just remove it, and then you won't get those structural characteristics expressed. So, yeah. But you can change messenger RNA concentrations. That's a great point to change the orientation of the development, right? So you can confuse and with the anterior, posterior, dorsal, ventral. I've seen flies before that will produce, wings on the ventral portion of the body, legs up on the dorsal portion. So you can change a lot of that orientation. So these homeotic selector genes, these are what really are fine-tuned to produce the legs and the wings and the mouth parts and whatnot. But you have several of these homeotic genes, and they start with the maternal effect genes. So keep in mind, as each one of these are expressed, what you're doing is it's coming from kind of a broad identity of those structures, and then narrowing it down or dialing it into the specific structures. And so you have maternal effect genes that regulate gap genes. Gap genes will regulate pair-rule genes as well as other homeotic selector genes. The pair-rule genes regulate the segment polarity genes, also homeotic selector genes, and then those regulate more of the homeobox or the homeotic selector genes. Okay, so again, what you're doing is you're getting positioning of anterior, posterior, dorsal, ventral positioning, and then you're starting to separate the insects into different gaps, and then different segments, and then different structures. Eli.
Are these genes kind of named after what they do?
In some cases, yes. There'll be like a phenotypic characteristic of them. I mean, the rule of thumb is if you identify it, you get to name it. So not always the case of what they do. So like Krupel, I don't know what Krupel does. I just know that it's a gap gene. Giant, I have no idea. Talus is probably, no tail, no abdominal segment. So yeah, it could be a variety of different things.
What berry gene does?
No, what does it do?
Okay.
Hunchback Western, they do look in that.
Yeah, so a lot of the phenotypic characteristics, you'll see it. Mm-hmm. Yeah, so like if you look at the the homeotic selector genes, I mean, the labial genes for the mouth parts. I mean, you've got abdominal A and B genes for the abdominal segments, antennapedia or the antennal segments. Ultrabithorax is one that's again that interconnection between the thorax and the abdominal segments. So this is only kind of a high level snapshot of the different genes that are involved. There are several more that play a role here. Awesome.
So I know the switchback, I'm aware that it's both maternally inherited as RNA and also as like a regular gene that's expressed. Why is that like basically the only one in that list that does that?
So with hunchback, caudal, nanos, and bicoid, is that what you're referring to?
Yeah.
Yeah, so I mean they've gone through and they've done, if you knock those out, or let's just say you reduce their expression, then you get an abnormally formed oocyte, right? So those are the key messenger RNA titers that produce those proteins to differentiate the polarity of the oocyte, right? And so through just different manipulations of decreasing or knocking down, if you will, the expression of those messenger RNAs, you can change that orientation. So, and then the others do play a role somewhat, but they're not as important as those four that I showed you in that graph. All right, any other questions? All right, so basically what we're doing is we're forming the egg, and the insect is expressing genes that start to differentiate the temporal and the spatial location of its different portions. So for embryonic development, so I'm not going to go into great detail on cell division. Everyone should know what mitosis meiosis is by now. But the oocyte is arrested during the metaphase of first mitotic division. and then it continues to mature after it's oviposited. So the oocyte will complete meiosis after fertilization. This results in a haploid nucleus, and then you have three polar nuclei. And these are what are fertilized by the sperm that enters that micropyle apparatus at the top of the egg. And so the different phases here of what happens, again, you have these partial divisions of cells, of these daughter cells that basically produce this ring structure, okay? And that's what's going to develop into or produce the embryo. So the zygote divides mitotically, it sorts out the cytoplasm into these daughter cells here. It's known as holoblastic cleavage. There's a large amount of yolk that limits the first cleavage, so it's considered more of a meroblastic type of event because the cells don't entirely cleave or separate. They're always intact. And then the daughter nuclei continue to divide and without formation of new cell membranes, but they form this syncytium here with the several nuclei, and that's basically what this structure here is, this syncytial structure, or this ring structure of these different daughter cells. And again, this is the orientate, or I should say, the starting point of the developing embryo. So when you have the insect oocyte here, you've got these energids here, these dark dots here, which are the daughter nuclei. They're surrounded by the cytoplasm, basically for nutrients. And they're not incorporated into new complete cells at this point. But what they do is they tend to migrate to the periphery, egg periphery, and then they'll continue to divide into different regions of the egg. They produce this syncytial blastoderm from those energids that move to the periphery of the oocyte. And then they also form these pulse cells from a few of the energids, which produce the germ cells, establish the germ line. And this is the origin of the Oscar gene that I mentioned in the previous slides for the anterior portion of that developing embryo. So just a different illustration of the energids that form the syncytial blastoderm, and that you have these yolk cells or vitellophages. They're non-dividing energids, but they do digest yolk, and then they start forming the midgut epithelium later on. So this cellular blastoderm here, this is basically membranes that develop around the different cell nuclei of the syncytial blastoderm. And then again, they're forming this cellular blastoderm. So you're starting to see the external structure of the embryo or the oocyte, and then eventually each of these cells start to form the different layers, and then you start seeing more of the structural features of that developing embryo over time. So you also have this embryonic primordium. It tends to develop into the embryo. It forms the germ band that represents the ventral region of the insect body. And then, as I mentioned before, you have these morphogenic gradients, these messenger RNA titers or concentrations that direct the determination and the differentiation of this developing embryo by activating or inactivating transcription factors or the transcription of genes for those anterior and posterior structures. So again, if you look at the developing embryo here, again, you have the energids here that start producing that blastoderm, and then eventually the embryonic primordium. You have the pulse cells here that start producing that Oscar signal for the anterior posterior portions. And then eventually you're starting to get the formation of that embryo in the egg. So again, what are the morphogens that differentiate the anterior portion and posterior portions of the insect? Yeah, so you got bicoid, Oscar, caudal, hunchback, right? Nanos as well. So as I mentioned before, what you start seeing is that you have this germ band formation that produces the overall structure of the embryo. So during gastrulation, you get the formation of multiple layers here that from the germ band cells, and they tend to migrate inward into the yolk. And then this gastrulation process here will produce a layered body of different cells that interact with each other to start differentiating the different portions of or segments of the body. So again, you have the germ band that forms, you start getting this gastrulation event. Here, you get this ventral furrow that starts to produce the mesoderm and the ectodermal cells for that developing embryo. So as I mentioned, for this ventral furrow here, it's formed by the germ band. It tends to elongate and migrate upward, forming the mesoderm. You have other genes that are expressed, for example, like Heartless regulates these mesodermal cell migration. Other genes such as Twist and Snail will divide the blastoderm into the ectodermal, endodermal, and mesodermal layers, so the three different germ layers of the developing insect. And then the germ band will extend to produce the trunk of the insect, whereas the ectoderm is on the outside and then the mesoderm is on the inside. So you're seeing these three levels or three cellular levels of development to start to form that insect. So here's where the homeotic selector genes or the segmentation genes start to really define the structural features of the insect. So segmentation starts with the maternal effect genes, and they activate or repress gap genes to establish segment identity. So the broad segments of that insect. Then you have bicoid, hunchback, caudal proteins. The patterns of these proteins or I should say the transcription patterns determine how the gap genes start filling in the gaps of those broad segments of the insect. So these gap genes, they'll regulate the expression of pair-rule genes, which divide the gap genes into parasegments. And these parasegments include 3 mouth parts. There are 3 associated mouth parts, 3 thoracic segments, and then 8 abdominal segments. All right. So as you can see here, it's going from kind of the broad strokes of that, what that insect looks like, and starting to fine tune it into the individual segments. You also have the expression of these segment polarity genes. So they're reinforcing that these repeated structures appear in each segment. Okay, so for example, the 3 thoracic segments having 2 legs on each segment. So it's reinforcing the expression of these structures on the segments. And then the gap and pair-rule gene interactions, they're regulating the homeotic genes that establish the identity of each segment and then the characteristic structures of why they look different across different species or genera, families, or orders of insects. Any questions? So, as I mentioned before, you tend to have these germ band cells that produce that ventral furrow, and they start to produce the mesoderm, the endoderm, the ectoderm. And so what you have here is you kind of have this broad structural difference between these cells, but they're kind of just floating around, right? They're not really producing any type of structure. And so what happens is the germ band will start to elongate and widen, and then it produces these amniotic folds here. And so what's happening is you're starting to get dorsal closure where the insect is developing inside this egg structure. So it extends from the ventral midline, emerges to form a double layer of extra embryonic cells. The amnion forms a continuous margin of the ectoderm, and then you have the serosa that's detached from the ectoderm from the germ band. And so during this, what this is called is blastokinesis. So the germ band during this event exposes new surfaces of the embryo to the remaining yolk and to be able to start again producing that structure. So you have the here it's just showing the formation of this amniotic and germ band invagination here to produce these different structures here. So again, you have the embryo, you have the germ cells that start to produce that ventral furrow. You get these amniotic folds, and then the embryo or the insect starting to develop inside that egg structure. And again, it's just going through several different types of remodeling of these germ layers, if you will, to produce the overall structural characteristics of that insect. So here in this Drosophila model, what it's depicting here is embryo movements during blastokinesis. And so the germ band movement tends to reverse the position of the embryo and the yolk, as opposed to the embryo position in the yolk. And then during blastokinesis, you have ectodermal growth over the dorsal portion, and it completes this dorsal closure with the yolk contained within the embryo. And during blastokinesis, again, it's displacing the embryo within the egg to enable it to utilize more yolk. And again, the yolk within there is providing nourishment, but it also has different morphogens being expressed to help with the full development of that insect. Any questions so far? Yeah, Bean.
Segment. So if you got
The which one?
the three thoracic
Mhm.
segments
Right.
existing, does that mean if you figured out how to make another thoracic segment express, you could have an insect with like 8 legs?
Yeah, absolutely. Right. They've done all kinds of manipulations with this model. And that if you get them, again, it's being able to capture spatially and temporally, the expression of those. But yeah, you could replace one of these homeotic genes with say a bithorax type of expression where you can get 4, probably even 5 segments, right? How functional is that insect after? I don't know, but I know that you can pretty much manipulate the expression of these different structures anywhere on that insect body by manipulating these homeotic genes. Eli.
I guess with this model, it's showing an adult Drosophila.
Mhm.
Is that with like egg development and embryonic development, is that the same for like pupal development? Is that would, I don't know.
No, that's a really good question. To my knowledge, all of these happen before pupation. So you have to capture all of these different events during the embryonic development. Now, during pupation, there is a whole nother structural reorganization, of course, right? Until to get to the adult structures. And again, you'll get different, it's not to say that there aren't other homeotic genes that are not later expressed, but a lot of these, the super important ones to get that insect built, for lack of a better word, happens during the embryonic phases. So, but it's not to say you can't manipulate pupal development down the road, right? So, for example, right now USDA APHIS has a grand challenges call for research proposals for New World screwworm. Okay, so the US is really worried about the movement up from Mexico, from Central Southern America on these screwworms to come in. Okay, so I don't know if you're familiar with how these screwworms operate, but basically it takes one female to find a wound in, say, cattle. She deposits hundreds of eggs, and those eggs hatch the larvae. They fall to the ground, they pupate in the ground, and they emerge as adults, right? So one of the strategies is, if you can't control it on the animal, how do you control it on the ground? Okay, and one of those is, can you produce a chemistry on the ground that moves through the pupal casing to be able to disrupt development of the pupae where it doesn't emerge as an adult, right? Um, and if you think about it, it is a grand challenge. How the hell do you do that, right? So it's quite complicated. Um, but with the screwworm, I mean, sterile insect techniques are highly successful in eradicating screwworm, right? But they're also looking for other interventions that they can use along the way because you want complete eradication. It's a really odd pest because it only takes one to cause major infestation, right? So it's not like a lot of other cattle flies that are in there in swarms, and that you're looking at large scale population reduction. You got to target that one female. They're storing. Yeah. So yeah, yes.
Is
So
it like tsetse flies where they only really mate once?
yeah. Yep. Yeah, it's a really interesting pest, and the devastation that it causes, but it's relatively fragile, right? Because it's just in low numbers until it produces the eggs. So all right. So as I mentioned here before, after blastokinesis, you start seeing several different migrations of cells to form specific structures, internal structures. So first you have the formation of the gut. And so these are well characterized to happen in this order. And it's also keep in mind that it's let's say gut formation starts first, and then later on you have nervous system formation. I don't want to say that gut has to be formed first and then the nervous system and so on. They're all happening at the same time, but they have documented that there is a step-by-step process, even though they're kind of happening concurrently, right? So during this formation of the gut, or after blastokinesis, gastrulation follows, and it starts to produce these internal organs, from producing new germ layers, and then also migrating these to different portions of the insect. And so the anterior and posterior cells tend to migrate inward to form the foregut, the midgut, and the hindgut. And then the foregut and hindgut originate from ectodermal cells, and with endodermal cells starting to form the midgut. And again, you have a variety of different genes and transcription factors that are expressed during these events. You have transcription factor expression, or these on and off kind of molecular light switches that determine formation of salivary glands. The proventriculus, ceca formation is from cell migration, and also homeotic genes that play a role in this. So I mentioned ultrabithorax before, and antennapedia. Again, these have pleiotropic effects. They're not just for thoracic structures or antennal structures, but they also play a role on the internal organs of the insect. And then you have these integrins, which have spatial expression activity. They tend to be receptors on cell surfaces that are mediating these cell-to-cell interactions, and then again, forming the different gut structures of the insect.
Also reduces sex phones.
I have no idea.
Just what it sounds like it should, do something not
It probably does. I mean, as I mentioned in the previous slide, you have these different segmentation genes that are expressed, and they all, I keep saying that they have these pleiotropic effects because they have more than one role, right? And so I'm sure that it not only helps with forming, salivary glands, it probably helps form reproductive structures, right? Um, in the order that it works that way, I don't know, Dean, but Eli.
So is the midgut being formed by it's being formed around the remainder of the yolk cells?
Yes. Yeah. So everything is starting to go from the outer edges into the inner edges to form these different organs, tissues and organs in the insect.
So would the embryo kind of surround the remainder of the yolk and kind of envelop it?
Yeah, so that's what it's doing is it's taking in it's basically what do you say maximizing nutrient acquisition by doing that, right? So you have all these free floating yolk, right? And so it wants to be able to encapsulate and take as much of that yolk as possible for nourishment. So
So this is probably just review, but the exoderm, since the hindgut and the foregut are both made of that, would you say that like everything that the exoderm is like made of is what is shed during like molts? Generally?
yes. So we'll get into this in the development. So on Monday, I think we will get into this a little bit more because we'll talk about chitin synthesis and then we'll get into molting a little bit more other than what we did in the signaling systems. Keep in mind that not everything is shed, right? So, I mean, in the internal structures are not all of the internal structures are being shed during molting. A lot of it is the exoskeleton, endoskeleton, that whole framework, right? Um, does that answer your question? I think, yeah. So not everything is
Just
being,
order, like,
yeah. That internal environment, yeah, it wouldn't be good to get rid of that, right? So yeah, so most of it's external. So again, during this formation of the gut, you have a variety of different homeotic genes, if you will, that are starting to form these internal organs structures. So in the hindgut, you get the hindgut formation. You get patterning, cell arrangement. That's regulated again by several different of these homeotic genes. Malpighian tubules or the excretory system that form at the midgut, and then hand out these junction transcription factors that help provide some resolution in how the Malpighian tubules look and how they function. And then again, the mature Malpighian tubules will have other genes that are expressed to be able to produce signaling pathways more for their functional activity. So I'll finish up here today, but again, really what I would like for you to walk away with from this lecture or this unit here is to have that conversational knowledge of how a developing insect has its spatial patterns. Okay, so what are the genes that are involved? Okay, we talked about hunchback and caudal, bicoid, nanos, and how the different concentrations will help form the anterior posterior portions, but then you also have Oscar and Kekon and Torpedo that form the ventral and dorsal functions. But at the end of the day, really, it's about there are specific morphogens or messenger RNAs that differentiate the broad orientation of the insect. And again, bicoid, hunchback, caudal, nanos for the anterior, posterior positioning of the developing insect oocyte. And then also thinking about how those segment polarity genes and homeotic genes can produce different structures. Again, they're highly conserved. They are expressed in all insects, but it's the expression level, the mutations that give the different structural features on different insects. Okay. Any questions? Ozzy.
How many of these genes, because I know that there's been research done on like hunchback specifically, do you know how many of the genes are conserved in other arthropods? Like how much of this is shared by the entire phylum?
We have the same genes. Yeah. So homeotic genes are conserved across all animals, right? So a lot of what we know about human embryonic development was defined in Drosophila tribolium models, right? So we mentioned earlier in this class, I mean the um, genetic makeup of Drosophila is like, I mean, almost 70+% of that of human genetics, right? You know.
Or what role do genes like segment polarity genes play in us? We don't really have segments in the same way that insects do.
Let's go back here for a minute. Yeah. I mean, I think it's 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 the same kind of situation. And like you have head, thorax, legs, right? Arms, legs. And I think that's those you could look at them as different segments, right? I would say that that would be the closest thing to it, right? There are all kinds of, deformities and malformations that we see in humans all the time based on, these differences in expression patterns or mutations associated with homeotic genes, right? And that's not just external, I mean, internally too. There's a lot of, just horrible diseases out there based off just dysregulation of these different homeotic genes, right? So hope that helps. But yeah.
We specifically have like these exact genes, like hunchback specifically, or is it like a like
No,
homologous?
it's a model. Yeah. So there'll be different names for animal embryonic, or I should say human embryonic development, right? Even for like mouse models, because a lot of the say Drosophila models have been translated over into mouse models and so on, right? So I would say that you would probably find homology associated with them. Whether they have the same name, probably not, but there would be some percentage of being homologous to, I don't know, tailless, right? So, any other questions? That was a good interaction today. Um, Let me know if you have any questions on the exam. Again, if you need a bit of extra time, that's fine too. It's on there as that Word document. So if you can, there are some questions from other people about when I say depict and design. For like depict, I was meaning if you can kind of draw like that monopolar insect neuron and get the structural features of it. If you can do it in Word form, that's fine too. I'll take that. A lot of you are pretty descriptive in using that. When it says design, I'm looking for a high level design of an experiment. You don't have to illustrate it or anything like that. Okay. If that's easier for you, that's fine with me too, as long as I can interpret it. Okay. All right. Enjoy the warmer weather. Stay well. Have a good weekend ahead. We'll see you on Monday.